Now we're going to cover data acquisition a little bit and talk about kind of the output from these mass analyzers. So from a mass spec, you would run a sample and you're going to get an entire raw file that's full of scans, and you would be defining those scans when you're setting up your mass spec run. But for example, you would have a single mass spec spectrum or precursor. This is an example of water, again, where you're looking at the mass to charge ratio, and this is looking at the different ions that would form from water from this example. So you have both single spectrums of intact masses, whatever those might be, and then you'll probably also define fragmentation or tandem mass spectrometry. So in many cases, just knowing the precursor mass is not enough detail to know how a compound goes together or a sequence of amino acids, for example. So in MS or MS2 or even MS to the N, but we're not going to talk about that too much in this lecture, there's selection and then fragmentation of specific ions. And then the fragments themselves are analyzed and read out in a spectra. So there's a few different modes of data acquisition or ways of setting up your LC-MS experiment. And so I'm going to talk about those now because it's dependent on what you would like to learn from your experiment. So multiple or parallel reaction monitoring is a precise, fast method which can provide absolute quantitation based on a known standard. A triple quadrupole mass spec is commonly used for this, and this might be useful in a setting where you're trying to quantitate a biomarker, or you are looking at a specific level of drug or specific post-translational modification. So it's targeted mass spectrometry. You need to know what you're looking for, and you have to have developed a method to look for it. But in this parallel or multiple reaction monitoring, there's first an ion selection for a precursor ion. So in the first quadrupole, only this blue square is being monitored for. Everything else that's not the blue square is being ignored. You will not see it in the data readout. And then in the second quadrupole, there's fragmentation of the blue square, and only individual fragment ions are being monitored in the third quadrupole. So this gives you really fast and very specific assay. And you would need to set this up with a known peptide or known standard. But yeah, it can be very useful for methods where you're going to quantitate the same things in many different samples, and you want to be able to do it as quickly as possible. For untargeted mass spectrometry proteomics, we are normally operating in data-dependent acquisition. And this means that the mass spec is performing a precursor scan. It's looking at all of the intact ions in that scan, and it is choosing the most abundant or the most interesting. There's a number of different ways to define what it takes from that precursor scan, and then selecting those for fragmentation. So, an example is here where you have, this is a smaller range of precursors, but it is selecting this window, and specifically this 452 is the main ion in this window. You can imagine, however, that the window of selection is user-defined, and you might include other ions in it. The mass spec does take, or you take note in the data analysis that there is interference or potential other things present in the fragmentation process. So this is an example of this ion being selected, fragmented into this MS fragmentation scan. And so it would go down, there's ways of doing it, either the top 10, top 20 most abundant ions. You can tell the mass spec to, ignore an ion that it's seen already. There's a lot of different methods and things you can play around with in the data acquisition method, but this is the most common data acquisition method for untargeted proteomics. There's also data independent acquisition, which is becoming more popular, in which an MS scan is performed and all ions within a selected M/Z range are simultaneously fragmented. So it's like what we had here, where but instead of just a small range of maybe 1 or 0.7 Daltons, we have a 20 M/Z isolation window. So here's an example. Here's how the isolation window looks for DDA versus the isolation window for DIA. And it just steps down the entire amount of range that you're looking in, and it's fragmenting all of these ions at the same time. So you can imagine that the fragmented data is going to look very complex. And this results in highly convoluted product ion spectra. And generally for the data analysis, it's quite complicated, and you would need to do a lot of runs prior to this in data-dependent acquisition and develop what we call spectral libraries. But it's becoming, there's software and techniques that are being constantly worked on to improve this because it is very fast. So those are the different acquisition modes. What you can do with those acquisition modes, I'm going to just chat briefly about different flavors of mass spectrometry. So top-down mass spectrometry is looking at an intact form of a protein. So there's no fragmentation or there's no digestion prior to adding your sample into the mass spec. So the whole protein is ionized. And I have here kind of the comparison of ESI versus MALDI. MALDI is often used for this intact mass where you only get a single or maybe up to a triple charged ion of a protein, and it's kind of this lower resolution, but can give you an intact mass. And electrospray ionization gives you these multiply charged species where you can see the full envelope of isotopes. And then you can fragment the isotopes and identify where specific modifications are occurring on a protein. And this is popular, especially with the lab I did my postdoc with. So obviously I had to bring it up because you can see when there are specific combinations of post-translational modifications occurring. And this is something that you can't as easily determine doing traditional bottom-up mass spec. But yes, there's a couple of really good papers you can read to learn more about that.
There are other applications for that are kind of cool to think about also, that include things like looking for covalent modifiers. So if you're working on drugs, because covalent modifiers have come back as a class of drugs that people are interested in again, this kind of waxes and wanes in the drug discovery field. But because of that, in addition to post-translational modifications, you could treat with a small molecule compound that's a covalent modifier, and then you could look to see what percentage of your protein is actually modified. So the quantitation for something like that in this is, I don't know what you think, Emma, but I think it's better than what you would get from bottom up, just because you're looking at the full molecule, so you get the full picture of what percentage of different forms are modified.
Absolutely. Yeah, that's a really good point. I had a whole project of that, where then ironically, the covalent modifier the chemistry lab had synthesized wasn't actually a covalent modifier. It like completely changed the structure of the enzyme. But we learned that through top-down mass spec. And another really important application might be looking at different splicing isoforms, where it might be really difficult to look at those through bottom up or to quantify those through bottom up mass spec. So bottom up mass spectrometry, which is most of what we're going to talk about the rest of the lecture, involves taking a protein or a mixture of proteins and digesting it with a protease. Trypsin is the standard protease into peptides, and then analyzing the individual peptides by mass spectrometry. You then have to do data analysis to match these peptides back to the source proteins. And obviously, any peptides that are either too long or too short, you won't be able to see by mass spec. So again, there's a lot of there's advantages and disadvantages to both. As usual, it just depends on your application. So top down, you're looking at an intact protein where you're not using any protease, and you're identifying proteoforms, is what they're called, which is combinations of PTMs on a specific protein. It does require that, so there's a few different ways, but you can't just run an entire helolysate at once. You won't be able to identify as many proteoforms. And it doesn't work very well for large proteins. So over 80 kilodaltons, you're going to be too highly charged, and it's the fuzziness that Dr. Mosley kind of talked about. And protein fragmentation is something that's still, being optimized and worked on. There's constantly more papers out, really some interesting things. The advantages of bottom up is that it is a more mature technique. This is what people have been doing the longest, and there's a lot of different software available for analysis. It's pretty quick, and a lot of the techniques have been worked out. The disadvantage is that you only see part of a protein. You're never, or very rarely would you get 100% sequence coverage. It can require using a lot of combinations of proteases and having pretty pure protein to begin with. There might be PTMs that are on peptides that are too long or too short, and you won't see those. And yeah, there's intense data analysis for both of these, is what I would say, is that mass spectrometry is a data-driven technique, and the protein inference problem, which is, determining whether a peptide goes to a peptide with homologous sequence in multiple proteins, which protein can that peptide be assigned to? And we'll talk about that a little bit later too.
And I'll maybe mention here that one of the things you might notice for both of them is that the sequence coverage and the need for purified proteins, that actually is a biochemistry challenge as much as it is a mass spectrometry challenge. So for any given protein, just like if you're going to do X-ray crystallography or some other type of cryo-EM or some type of structure analysis, having extremely pure protein is really important to do any of these types of analysis if you want very high sequence coverage. And so it's a challenge for actually all of these, and we do spend a lot of time in the core working with people on their purification methods, partially because that is something that's really a requirement for certain types of mass spectrometry data, especially for things like full coverage post-translational modification mapping, which is something that we work on quite often. So just thought I'd mention that.
Yeah, absolutely. So how do you fragment your ions or your peptides? So you have your intact ions, you have donut precursor scan, and you can see them. You're going to send some of them for fragmentation. So what does that mean? And there's a few different types. I'm not even going to talk about a couple of them. I'm just going to talk about the most commonly used ones here. And the most common is this collision-activated dissociation, collision-induced dissociation, higher energy collision dissociation. As you can tell, it all mentions collision dissociation of some sort. And that's the example here at this top section of this fragmentation scheme. And essentially, after trapping your ions and selecting a precursor ion or segment of ions, you increase the kinetic energy in whatever portion of the mass spec you're using to fragment, and you allow enough of a neutral gas to be present that the ions collide with themselves and the neutral gas and fragment into these specific B and Y type ions. So you can see just the kinetic energy causing this amide bond fragmentation. And these are the types of ions that you're getting from that. So then this is what you look for in the fragmentation scan. The other most common type of fragmentation scheme is electron transfer dissociation or electron capture dissociation. So in this case, there's some reagent that is added instead of the neutral gas like we're using for CAD or HCD. There's some anionic small molecule reagent that creates radical bond breakage and creates these C and Z type ions. It's breaking this N-C alpha bond, and both even and odd electron product ions are produced. And, you know, in a perfect world, you would break every peptide bond, you would break a bond between every amino acid in the peptide that you were trying to fragment. This would be a perfect world, right? So this is an example of a CAD MSMS fragmentation. And so you can see, this is how I mentioned the B and Y type ions that are being created. And ideally, you would see both B and Y ions that match specific amino acid residues. And this is essentially peptide sequencing, where this population of peptides or proteins subjected to fragmentation, but the mass spec knows what the major ions were in the first scan, and the type of fragmentation used determines what type of ions you're looking for in the fragmentation scan. And in this perfect fragmentation world, you would get this ladder of ions where you're sequencing every piece of the peptide that you started with, if that makes sense, from the B and the Y side. And it's very rare that you would actually get to see all of these. You're going to see some number of these. And that's going to give you a clue that's going to let you determine how confident you can be in the identification of this sequence of peptides.